duplex sequencing be used for outside genetic toxicology?
Okay, so the technique can have uses in a variety of fields, say oncology, where you need to detect a rare mutation event. So perhaps in oncology, you might want to look at the detection of residual disease after a patient has been treated, or perhaps you're interested in looking at the increase in the number of drug-resistant populations before that is evident in a medical setting. You could also perhaps use the technique for looking at CRISPR-edited cells, for looking at on-target or off-target editing events. The high sensitivity of the technique is very useful here for detecting rare off-target events. I believe TwinStrand gave an excellent presentation on Friday, which is available on demand, which I'm sure will go into a lot of these other applications in more detail. So if you're interested, please go and watch that talk.
Brilliant, thank you very much. And we also have Dennis Wang, Associate Professor and Head of Bioinformatics, University of Sheffield and A-Star Singapore, joining us today. Welcome, Dennis. Great. So the next question, Matt, is for you. And is the following. In what circumstances can we get away with using expression-based cancer cell transcriptome identification?
Yeah, I guess the main circumstances are when you have some strong prior biological knowledge about the particular tumor type. So the example I gave of renal cell carcinoma, while the results of the expression-based annotation were potentially misleading, I don't think anyone in practice would actually be misled about that, because the biology of the tumor is well enough understood that you can sort of sanity check and correct the results that expression-based annotation give you.
Great, thank you very much. Dennis, welcome. We can see that you are on camera now. The next question is for you. You were presenting on unsupervised learning for personalized medicine. You touched upon the challenges and applications within your presentation. If you would need to pick one challenge that you consider as a key challenge, which one would that be and why?
I think, I had many challenges in the presentation, but probably one of the key challenges that's influencing or has a lot of attention for the machine learning community is kind of explainability of the models. So some of these machine learning methods are very powerful. They can get great prediction accuracy or diagnostic, have great diagnostic utility, but in order for like clinicians and healthcare professionals to trust some algorithms in practice, they need to be able to understand how the algorithm works, how it comes to prediction, and also those algorithms need to be somewhat intuitive for the patients as well. So I think that's one of the main challenges. These algorithms are becoming very complex. They're learning from a lot of data to make very informed decisions. But how they work isn't as easily communicated to the end user.
Brilliant. Thank you very much, Dennis. And now back to Anne. What kind of mutations can be detected by duplex sequencing? Is it only single base changes?
No, you can detect bigger mutations. You ought to think about your NGS sequencing read. So you can detect multiple base changes and small indels that can be incorporated in the length of a read. But it is possible to detect translocations if you have the translocation on each half of the gene, if you've got the appropriate controls in place. So it isn't just single base changes that can be detected.
Thank you very much, Anne. And now over to you, Matt. Can cancer transcriptomes be identified without whole genome sequencing?
Yeah, I mean, you could in theory. You could certainly get away with whole exome sequencing or doing copy number arrays. In fact, in a recent paper, we use copy number arrays to genotype things. I sort of generally think that the cost of whole exome or genome sequencing now is such a small fraction of the cost of expensive single cell experiments. There's not a huge rationale for trying to get away with avoiding that cost, but it is potentially possible, yeah.
Brilliant. Thank you very much. We still have a few minutes. Please feel free to post your questions in the chat box, and our speakers and Matt and Dennis will be happy to answer. Great. Now we move on to Dennis. Dennis, so the area of genomics, genomic medicine, clinical genomics has evolved quite rapidly over the past few years. How do you think this area is going to evolve further in the future? Are there any specific topics of interest that you and the group are kind of like looking into within this space?
Yeah, I think with these kind of very powerful technologies to collect data, like sequencing technologies that are becoming cheaper, there's kind of quite a few kind of applications that could change the way we diagnose and treat patients. One of the kind of most interesting for my group is kind of looking at heterogeneity of the tissues. Right, like a lot of diseases, from metabolic diseases to cancers, cell types change, so a given tissue is we can use sequencing to understand how specific cell types change due to a disease, whether a particular tumor, how it evolves over time during treatment, or a particular tissue like placenta, how it changes as pregnancy takes place, and then I think generally also what's exciting is kind of longitudinal profiling. So a lot of the early genomic studies are kind of just snapshots of a biopsy tissue, but if you can kind of track tissue as it changes over time, you can follow how treatments, how the patient and the disease responds to treatments.
That's great. Thank you very much. What about you, Matt? How do you think the field is going to evolve in the future, and what areas are going to be of key importance for you and your group?
I don't really know. I'm not going to prognosticate on the future, to be honest. I'd be interested in asking the other speakers a question if there are. If there's time to do that, but I'll let you go through your questions first.
Great, thank you very much. Anne, what about you?
So it's trying to see the advantage of this technique. So in our field, we have a lot of really good, robust assays that they all rely on reporter genes for looking at mutations. But with error-corrected sequencing, we can get information directly from the genome and get not only mutation frequency, but mutation spectra. So this can provide an additional biomarker for the carcinogenic risk that is present by looking at the mutational spectra. And there's a huge advantage of this technique in that you can use it in vitro or in vivo in any cell type, in any animal type. You don't need to have a transgenic rodent or anything that's a bit more complicated. You can apply it anywhere. And so perhaps, I could talk for hours on this, but perhaps an example that's easy to understand is if you were to take tissues from a rodent study that wasn't designed to look at carcinogenic risk, but used your high sensitivity error-corrected sequencing on a particular tissue harvested, then maybe that would give you the same level of prediction of human cancer risk as running a carcinogenic study, which in rodents lasts for 2 years. So you could be reducing your timescales for getting that information, but more importantly, reducing the number of animals you need. And there are many other exciting possibilities in the future, but with the interest of time, I will halt it there.
That's great. Thank you very much, Anne. Brilliant to hear from all of you. Great. We still have a few minutes. Please feel free to post your questions in the chat box. Otherwise, yeah, we will wait a few more minutes for the questions to arrive. How did you enjoy the morning presentations, and Matt and Dennis? Have you had a chance to see each other's talks?
Yeah, so I'm just going to ask Anne a question. So I was super impressed by the cosine similarity between your the different dosages, where essentially you're able to really precisely define what the mutational profile of compound is. I was wondering if you thought about or are looking in existing tumors and taking that really tightly defined profile to see if you can see evidence of any particular compounds driving cancer risk.
That's an excellent question. Personally, no, we are not doing that, but there is data out there in the literature, or data that's starting to be formed. We are a small part of an international group that's looking at this, and we will be comparing our data with all the rest of the data generated around the world to look exactly at that. But personally, no, we are not doing that, but that would be super, super interesting, and it's definitely something we want to do. So yes.
That's great. Thank you very much. And do you also have any questions to Matt or Dennis to Matt and Anne?
I can ask Dennis a provocative question if you like.
Okay.
Please feel free to go ahead.
So Dennis, I was really...
We should talk about this at the bar. Too bad we can't be in person. Yeah.
So I was really struck by your comments of one of the limitations of the machine learning approaches being the lack of in the drug prediction context, being the lack of a clear target to optimize on, a clear variable to optimize on. Does that sort of doom the Google DeepMind AI sort of thing to fail, because without that to guide the deep learning target identification?
Yeah. Maybe. I mean, I'm not too familiar with the overall goal of I know they just recently spun out a company to kind of further the translation of that technology to drug development. But I guess with the DeepMind, I mean, they had a very specific goal, right, to solve a protein structure, and they had very good validation data, right, from exactly. X-ray crystallography and so forth. So it was they did it in the context of a machine learning challenge where there was already kind of gold standard references to benchmark against. But I guess for discovery, there isn't that kind of gold standard, right? I think I mean, one of the other challenges is just producing reliable target variable data for machine learning. So whatever specific outcome you're trying to predict. So in my case, I was just showing some examples where it's very hard. Even there's a lot of uncertainty in the IC50 value, right? In an in vitro setting, which is actually should be quite controlled, and you should get pretty reliable measures. But even there, you're gonna get some uncertainty in the IC50s. So then if you're trying to do this on animal models or even humans, it's gonna be even more difficult to get reliable measures of your target variable to train your algorithm.
So should we be spending more money on doing good experiments and generating good data than on methods?
Yeah, I think so. Yeah, I think that, I mean, these algorithms are still very kind of data hungry. But even if you're using kind of unsupervised methods, you still need to identify a very clear goal for the algorithm. I'm not sure, what is your take on that in kind of the single cell research, because I'm trying to get into the single cell research, but I know there's a lot of machine learning development and, you know, using single cell data, but does this problem also occur?
I mean, definitely. I mean, there's always this issue of like, what do you use as your test data set, and unless you've got a really, you know, nice, well-controlled test data, then it's hard to say anything meaningful about it. I guess the advantage there somewhat is because it's such a new field, you know, going from iterative improvements on existing tools has sort of marginal benefit, but when there's no tool for a specific application, then, you know, sort of anything is a step in the right direction, even if you can't validate it 100% perfectly.
Yeah.
Brilliant, great. Well, thank you very much for all of you to join this live Q&A session. It was really nice to see you all, and I really hope that you enjoyed the morning talks, and you will enjoy the rest of the day as well. We will have now a short morning break, and the presentations will continue from 10:40. Thank you very much once again, Anne, Matt, and Dennis, for joining us today, and have a lovely rest of your day.
You too.
Thank you. Bye.